The gut microbiome is the complex ecosystem of microorganisms in the gastrointestinal tract and has emerged as a key determinant of human health and disease. Previous studies revealed that sex-specific variations in the gut microbiome, referred to as the microgenderome, affect immune responses and susceptibility to disease. However, these studies explored the microgenderome using stool sampling, which is not representative of the entire gastrointestinal tract. Because the small bowel plays key roles in immune responses and metabolic health, exploring the potential influence of sex-based differences specifically in the small bowel microbiome may assist in the development of personalized therapies and microbiome-based interventions to enhance human health. The Reimagine study is an ongoing investigation aimed at understanding the role of the small bowel microbiome in human health and disease, utilizing samples obtained during standard of care upper endoscopy without colon prep. Reimagine study subjects with available small bowel microbiome data were selected for analysis, matching for age and BMI, to minimize variability. The resulting final cohort comprised a total of 266 subjects, including 133 females and 133 males. And here are the results. Duodenal microbial alpha diversity was not significantly different between females and males, which aligns with findings from previous stool microbiome studies. In contrast, beta diversity differed significantly between females and males, indicating that duodenal microbiome composition was significantly different between the two groups. No significant differences were identified at the phylum level, but sex-specific differences were identified at the family and genus levels. Genera such as Faecalibacterium and Rhombutia were more prevalent in the female duodenal microbiome, whereas Sarcina and Lactococcus were more abundant in males. Analysis of the duodenal core microbiome also identified significant differences between female and male subjects. Specifically, the genus Granulicatella was the fourth most prevalent in the core microbiome of females, but was less prevalent in males, whereas genus Leptotrichia was found in the core microbiome of males only. But what do these microbial differences mean functionally? To determine this, we performed an analysis of predicted microbial metabolic pathways in females and males. Despite the number of significant differences at the genus level, only 16 microbial metabolic pathways were differently enriched between males and females. As metabolic pathways can also be driven by community-level changes, we next examined duodenal microbial communities via network analysis. Microbial network analysis allows us to characterize interactions between microbes, which help drive the function and stability of a given ecosystem. 5 distinct duodenal microbial communities were identified in females and 6 in males, shown here in different colors. Interestingly, the lead genera in the specific communities also differed in males and females. Of note, the duodenal microbial network in females included a unique microbial community, shown here in orange, that was primarily shaped by the genera Streptococcus, Granulicatella, Escherichia shigella, and Rothia. In addition, this unique microbial community accounted for at least 5 of the different microbial metabolic pathways that were altered in females versus males. These pathways include bile acid deconjugation, which converts primary bile acids into secondary bile acids, commonly leading to diarrhea, and fructan biosynthesis, which produces complex carbohydrates that can contribute to digestive symptoms, particularly in conditions like irritable bowel syndrome, which is more common in women. Lastly, although there were more microbial communities in males, the overall network diameter in females was looser than in males. This was because of lower interconnectivity between microbes in the microbial network in females. As a result of this lower interconnectivity in females, network eccentricity was significantly higher, potentially suggesting greater susceptibility to environmental changes. In summary, the core duodenal microbiome differed significantly between the sexes. Genus Granulicatella was more prevalent in females, whereas Leptotrichia formed part of the core in males only. A unique microbial community in females that included Streptococcus, Granulocytella, Escherichia shigella, and Rothia was associated with differences in several predicted microbial metabolic pathways, like bile acid deconjugation and fructan biosynthesis. Microbial network analysis revealed differences in the number, composition, and interconnectivity of duodenal microbial communities in females versus males, with higher network eccentricity in females that may reflect greater susceptibility to environmental fluctuations. Understanding these differences may help elucidate sex-specific differences in human health and disease.